Hello, everyone, and thanks for coming. My name is Scott Taylor, and I'm an assistant professor at the University of Colorado in Boulder in the Department of Ecology and Evolutionary Biology. And it is a really great pleasure to be able to have the opportunity to share my work with you today. I was an undergraduate student in the wildlife biology program at the University of Guelph, and I actually grew up just a couple hours away from Guelph, so relatively local, and did my PhD at Queen's University in Kingston, Ontario, where I think in my second year, I actually co organized the uh, Ontario Ecology, Ethology, and Evolution colloquium that year. So it's really nice to be back, and I wish it was in person, but I'm still happy to be able to share some of our research with you digitally. So, this question is a question that has really motivated me since I was a kid, but I didn't know how to say it this way. But generally, I'm interested in the generation and maintenance of biological diversity. As a child in the forests of southern Ontario, looking at spotted salamanders and redback salamanders and eastern newts, I was really curious about where all that diversity came from. And now, as an assistant professor, and previously as a postdoc and graduate student, I've been asking and addressing this question mainly using avian systems. In order to answer the question, which maybe is unanswerable, but at least we can start to piece things together, I think about it in a framework that Rick Harrison laid out in Evolution since Darwin, wherein populations can be split by extrinsic barriers like mountain ranges or rivers leading to isolated populations, within which drift and natural selection can act to cause populations to diverge. At some point during the divergence process, you might sample a specific region of the genome, denoted here on the left as a phylogeny, and you might find that species 1 differs from species 2 at that site, but maybe not at other sites in the genome. Maybe in other places they're still similar. We could consider this to be phylogenetic species. As divergence continues, you get the evolution of intrinsic barriers, things like low hybrid fitness, hybrid sterility, those sorts of things, to the point where you might consider your diverged populations to be biological species, where there's some form of reproductive isolation established between them. At kind of the quote unquote end of the process, you might have what you could consider genealogical species, where you have exclusivity across the genome. So the phylogeny here is depicting that regardless of where you sample in the genome, species 1 is different than species 2. And I'll say most of the systems I work on aren't this way. And you do find a lot of shared variation, and that's partially why we work on them. You can flip this on its side and think about it as a continuum, a speciation continuum, from weak to strong divergence. And it's important to note that this continuum isn't directional. You don't have populations always moving in one direction. They can reverse directions, they can go back and forth. Certainly, speciation and diversification is context dependent, and ecology and environment can really influence it. But we can use multiple approaches to investigate diversification at various points along the speciation continuum. And that's what I'm doing as a grad student, as a postdoc, and now as an assistant professor, working on avian systems that fall out in different places along this continuum. Here pictured are red-poll finches, blue-winged and golden-winged warblers, which we'll talk a lot about, and mountain and black-capped chickadees. And along the continuum, you can see an image of clip art chromosomes that show where the genome is shared and where there are divergent regions, the divergent regions being indicated by an asterisk. So when populations are weakly divergent, you might expect most of the genome to be shared with only a few regions different. And that increases as you increase in divergence. So before we get into the talk, I also want to go over some definitions of words that I'll use, because I know not everyone here thinks about all the things I think about. So hybridization is a term I'm going to talk about a lot because we study hybridization in my lab. And when I use that term, I'm talking about the interbreeding of individuals from populations or groups of populations that you can distinguish on the basis of one or more heritable characters. This is a definition originally developed by Woodruff and then modified again by Rick Harrison in 1990. For those of you that are more visual, here's an example. So here you have the offspring of a zebra and a miniature horse inheriting stripes from one parent and small status from another parent. And these examples of hybridization, ones we create, are kind of the ones we think of first, I think. Think of a liger as well. And these don't occur in nature. The reality is, hybridization is common in nature. And a recent review that myself and Erica Larson at the University of Denver completed looked at how our understanding of the prevalence of hybridization in nature is increasing as we have more and more access to whole genome data, where we're finding in systems where we didn't expect there to be hybridization quite strong signatures, including in the history of elephants, as well as humans. So most of us have some proportion of our genome that is derived from Homo neanderthalensis. So hybridization is common in nature, and that's partially why we study it. Another term that I'm going to use is introgression. And so if hybrids are viable and fertile, so if they can reproduce themselves, you can get the movement of genes from one species to another by recurrent backcrossing. So here in this swordtail example, you can see that the first generation hybrid is 50% one species, 50% the other. But if that individual only ever breeds with individuals with swordtails, maybe because of a preference for that phenotype, eventually you get individuals that have a large proportion of one species genome and a tiny proportion of another. This is called introgression. When hybridization occurs on the landscape, we call those regions hybrid zones. They're regions within which genetically distinct groups of individuals interact and produce some offspring of mixed ancestry. And there's lots of different kinds of hybrid zones. There's ones maintained by tension between dispersal and selection. There's others that we consider mosaic, and there's even some where we think hybrids do better, bounded hybrid superiority zones. That said, hybridization in hybrid zones are really cool because they're these natural experiments that we could never really conduct in the lab that can help us understand the genetic basis of phenotype. So by having a lot of natural crosses happening out in the wild, you can look at homogenized genomes for regions that remain different despite hybridization, as has been done for Darwin's finches, where they were able to pinpoint regions of the genome related to bill size and shape, which have important implications for reproductive isolation because they influence song. And you can also use hybrid zones to understand the genetic basis of species barriers, which is a large focus of much of my work on chickadees. And I won't be talking about the species barrier aspect of hybrid zones as much today, but I will revisit some of our chickadee work at the end of the talk to give you an idea of where we're going. So the talk can be split up into 3 parts. The first is about warbler coloration and migration. The second is about asymmetric introgression of color. And the third is a study of color and reproductive isolation. And as we move from part 1 to part 2 to part 3, we'll move along that continuum of strength of isolation between the hybridizing species. So the first part of the talk, like I said, is about warblers, their coloration and their migration. And this work would not have been possible without David Taves, who's an assistant professor now at Penn State University, as well as Alan Brailsford, Bronwyn Butcher, Rachel Vallander, Philip Messer, Irby Lovett, Henry Strebe, and Gunnar Kramer. So if there are any birders watching the talk right now, you'll be familiar with the North American wood warblers. Certainly right now, the forests around Guelph are filling up with these brightly colored songsters that are neotropical migrants from Central and South America up to their breeding grounds in North America. The cool thing about this continental radiation is that unlike Darwin's finches, if you took the feathers off these birds, you wouldn't be able to tell them apart. There's very little morphological variation between the wood warblers, but there's a lot of plumage variation. And the extent of plumage variation in this group has led people to hypothesize that potentially plumage is involved in the diversification of the group. And you can see, too, that certain different species share plumage patches, like the black throat, which you can see on a number of species pictured here. So obviously, understanding the genetic basis of these plumage differences might help us further clarify the evolutionary history of the group. And we've been working in a hybridizing pair of wood warblers to try and understand the genetic basis of plumage variation. A really fun aspect of this project is that it's a mystery that begins about 183 years ago with John James Audubon, who wrote a letter to his friend Bachman, where he said, I suspect blue-winged and golden-winged warblers form only one species. This is pretty interesting because in 1835, nobody knew that these two species hybridized, and they look different and they sound different. So it wasn't until 1874 that the Brewster's warbler was described as its own species, and it wasn't until 1913 that anyone realized that Brewster's warblers were hybrid offspring of golden-winged and blue-winged warblers. So it's pretty interesting that Audubon had this suspicion well in advance of anyone knowing these birds hybridized. 76 years after he originally suggested blue-winged and golden-winged warblers were possibly the same species, this is when we figured out Brewster's warbler was a hybrid. So when people realized that these birds hybridized, they were really interested in plumage inheritance, and in looking at birds in museum collections, found that there was a lot of variation, pictured here in a painting by William Dilger. At this point, people recognized that the black mask and throat of the golden-winged warbler was either there or not. There was never an intermediate stage of that plumage, and it led Nichols and Parks both to suggest that it was a Mendelian recessive trait. The yellow body color, on the other hand, could be there, could not be there, or could be there in different shades. And so, although some people thought it could also be a Mendelian recessive trait, there was less support for that. Following attempts to understand plumage inheritance, people got really interested in why these birds hybridize and the ecology of hybridization. This took place prior to the availability of genetic resources. Interestingly, a lot of this work took place really close to the lab of ornithology, where Dave Taves and I actually produced the data that I'm going to present to you today. The current distribution of blue-winged and golden-winged warblers in North America, their breeding distribution is depicted here. Golden-winged warblers typically breed at higher latitudes and elevations than blue-winged warblers, but they really don't have a classic hybrid zone. They have more of a mosaic hybrid zone. And the breeding habitat that they utilize is secondary growth forest. They like scrubby habitat, regrowing orchards, places that are quite modified. And keep that in mind because we're going to talk about kind of the conservation context of the system shortly. Importantly, the range of each species is dynamic and tracks appropriate habitat. So here you can see the potential ancestral breeding range of the blue-winged warbler, and then the year the first specimen was collected across various places in eastern North America. And you'll notice that in Ithaca, New York, at the top right part of the range of the blue-winged warbler, the first collection wasn't made until 1970. So clearly, these birds are tracking the appropriate breeding habitat, which results in extensive dynamic and mosaic hybridization. So wherever they co-occur, they hybridize. And they co-occur in kind of patchy ways related to where their breeding habitat exists. Now, the breeding habitat that they use, like I said, has become more common, specifically after European settlement of eastern North America. Because of that, people assume that hybridization between blue-winged and golden-winged warblers isn't a good thing, and it's our fault. So because we created more breeding habitat, we're the reason that blue-winged and golden-winged warblers hybridize, and we should solve that by habitat management practices. We're going to revisit this later because this would suggest that the birds have only been hybridizing for about 300 years. So we'll come back to this. Early genetic work in the system found that you could differentiate two mitochondrial types, one considered a blue-winged type and one considered a golden-winged type. But really importantly, you can find the golden-winged type in a blue-winged plumaged individual, and you can find the blue-winged mitochondrial type in a golden-winged plumaged individual. So although this level of mitochondrial divergence would suggest a million years of isolation, contemporarily, there is no strong structuring or co-occurrence between plumage and mitochondrial type. This led people to look for genetic differences in the nuclear genome, and they didn't find any. Using allozymes, microsatellites, introns, AFLPs, or even a 16,000 locus dataset, which was generated by myself and Dave, didn't find any differences between the species. And this is pretty incredible because they obviously look very different, they sound different, they have different ecologies, and like I said, the level of mitochondrial divergence that they possess would suggest that they've been isolated for about 1 million years. So we took advantage of recent advances in sequencing technology to sequence 20 individuals, 10 that looked like perfect blue-winged warblers and 10 that looked like perfect golden-winged warblers. And we did this from a region wherein we knew there was a lot of active hybridization, hoping to take advantage of the fact that hybridization would reduce the genetic background between the birds we were sampling and allow us to detect regions of the genome that may be responsible for the phenotypic differences that they exhibit, like the black-throated mask or yellow body color. We aligned our genome data to a genome of the myrtle warbler and ended up with relatively shallow coverage and about 11 million SNPs. A SNP is a single nucleotide polymorphism, so one difference. You can think of it as one base changing to a different base. If you think back to the previous data sets, 16,000 was the largest data set that had been used, and now we're using a data set of 11 million. Not surprisingly, most of those 11 million regions aren't different between blue-winged and golden-winged warblers. In this plot, you're seeing FST, which is a measure of differentiation on the x-axis, and frequency, so number of SNPs on the y-axis. You see that most of those 11 million SNPs hover around 0, which means that that region, blue-winged and golden-winged warblers do not differ. But if you zoom in on the tail of the distribution, we do find regions of the genome that are quite different between blue-winged and golden-winged warblers, some of which are fixed, that little blip around FST of 1. We can visualize this differently in a way this is called a Manhattan plot, where along the x-axis you have relative position in the genome, and along the y-axis you have FST, that measure of differentiation. It's important to note in this plot that FST, the average, so the mean weighted FST between the two species is 0.0045, so very low. But there are 11 million dots on the plot, which makes it look elevated. Regardless, when you statistically look for the tallest buildings in this Manhattan skyline, there are only 6. And so it's not surprising that using 16,000 loci, we didn't hit these needles in a haystack. And not only are there only 6 divergent regions, they're also quite narrow, which again helps us understand why previous attempts to find nuclear genetic differences between blue-winged and golden-winged warblers failed. So we can then ask, what is near these divergent regions? With the assumption being that if these are the only divergent regions between blue-winged and golden-winged warblers, perhaps they exist near genes that influence phenotypes and might, say, code for color variation or pattern variation in plumage. And I remember when I first examined what these regions were, I was at home on a Sunday afternoon using a BLAST algorithm to identify these regions, and it was a very exciting afternoon. We'll zoom in on each of these peaks and talk a bit about what's around them. This first one is directly upstream of follistatin, which is a gene involved in feather bud development. And in the picture here, wherever it's dark, that's where follistatin is being expressed during feather bud development. This divergent region is directly upstream of ectodysplasion A, or EDA, which is involved in feather tract development and perhaps more famously known for its involvement in three-spine stickleback lateral plate morphology. This divergent region is also near another important developmental gene, wingless related integration site, or WINT, which is involved in feather morphogenesis. Again, you can see where ectodysplasion A and wingless related integration site are being expressed in these images of developing feather tracts. This divergent region is directly upstream of agouti signaling protein, which is known to be involved in dichromatism in chicken feathers, but more famously known to be involved in color variation, mouse coat color variation, and beach-adapted mice. Agouti signaling protein is in the melanogenesis pathway, and so perhaps it has a connection to the black throat coloration we see in golden-winged warblers. And finally, this divergent region is directly upstream of beta-carotene oxygenase 2, which is a carotenoid metabolism gene, and importantly, the yellow coloration in all of the wood warblers is a carotenoid-based pigment, and beta-carotene oxygenase allows the metabolism of ingested carotenoids prior to deposition in feathers. So this is very exciting. 4 of the 6 divergent regions are really close to genes related to plumage in some way, whether it's pattern or coloration, which was very, very, and continues to be surprising. We can zoom in on these regions to better characterize the variation around the gene. because we might expect that the variation would be within the gene itself, but we don't find that at all. For every one of these genes, as you can see here with follistatin, the divergent region is directly upstream of the gene of interest. So here, every gene of interest is in red, the exons in dark red, the introns in light red. The direction the gene is read during transcription is shown by a red arrow. And you can see that the divergent region for follistatin falls directly upstream of the gene, potentially in the promoter or enhancers for that gene. Similarly, the divergent regions for ETA and Wnt fall directly upstream of the genes, again, potentially in regions where you would find promoters or enhancers. The same is true for agouti signaling protein, and here we've mapped where the chicken promoters would fall in the warbler genome, right within this divergent region upstream of the gene. And finally, for beta-carotene oxygenase 2, the divergent region is again directly upstream of the gene. Importantly, for none of these genes are there any coding sequence differences. So none of the exons have SNPs. It's always SNPs directly upstream of the genes. And so perhaps you can imagine that these differences, these potential regulatory differences, can turn different parts of the genome on or off, influencing the plumage pattern variation that we see between golden-winged and blue-winged warblers. When we stepped out from those 20 individuals for which we found this pattern and genotyped 340 some individuals at these 6 divergent regions. For those individuals, we also scored plumage. And when we look at correlation between those genetic scores for those 346 individuals and plumage scores, we see an almost perfect correlation, which suggests to us that we really have found the genetic regions that underlie plumage variation between blue-winged and golden-winged warblers. If you think back to plumage inheritance and that black throat, a prediction that you would make then is that if there is recessive inheritance of the black throat, that in order to have the black throat en masse, you would have to have 2 golden-winged alleles at a given genomic region. And if you didn't have the throat, you could be heterozygous or homozygous for the other allele. And when we look at observed agouti genotypes, we find exactly that if you have 2 copies of the golden-winged warbler agouti allele, you have a black throat en masse. But if you're heterozygous or homozygous for the blue-winged allele, you do not. So this is really good evidence that the variation we find in agouti signaling protein is responsible for this pretty significant plumage patch variation between blue-winged and golden-winged warblers. For the birders in the audience, you might be thinking, well, what about Lawrence's warblers? Maybe you weren't because you don't know birds, but that's okay. The Lawrence's warbler that we have in our data set, there's only one. It is the only individual with blue-winged beta-carotene oxygenase 2 that's homozygous for that, and also homozygous for the golden-winged agouti allele, again suggesting that we really have found the genetic regions that underlie color variation in these birds. And it's hard to convey how exciting this is because in non-model systems, up until recently, it's been really hard to link genotype to phenotype. And so now to be able to do that within a system where we know there's similar plumage patch variation across the whole warbler radiation is really exciting and might allow us to better understand diversification of the North American wood warblers from a plumage perspective. We were also interested in trying to understand whether there's a genetic basis for migratory differences between golden-winged and blue-winged warblers, which do differ in their migratory pathways. In this picture here, you can see geolocator tracks from work that Henry Strebe and Gunnar Kramer conducted on golden-winged and blue-winged warblers across much of their breeding range. You can see that the Appalachian golden-winged warblers in darker orange go to northern Venezuela, whereas golden-winged warblers from the Great Lakes population, or blue-winged warblers, almost exclusively go to Central America. But there are some individual blue-winged warblers that go to South America, and then golden-winged warblers from the Appalachians that go to Central America. And so we asked with the same data, we asked with a larger data set actually, 77 individuals for which we had whole genomes and migratory tracts, whether there was a genetic basis for migration differences. And we found that regardless of plumage, just based on migration, there is a genetic association with where a warbler spends the winter. So if a warbler goes to South America versus Central America, regardless of its plumage color, we find this divergent region within which we also see big difference in Tajima's D, which is a measure that allows us to infer that there's probably been selection on this region. And different than the plumage results, there are differences in exons within this gene VPS13A. We know that this gene is involved in mitochondrial disease in humans, but we don't know its function in birds. But it's an interesting potential candidate for migratory differences between golden-winged and blue-winged warbler, well, between Central American and South American migrants. And we're continuing to study this to try and understand what this means. I think a big question then becomes why we could use natural admixture to detect the genes that underlie plumage and migratory differences in vermivora warblers. So if you think back to when I started setting up the system, I told you that the assumption is blue-winged and golden-winged warblers have only been hybridizing for about 300 years, since large-scale clearing happened in eastern North America. But when we use the site frequency spectrum and a modeling program and test several demographic models in order to estimate the timing of migration and onset of migration between blue-winged and golden-winged warblers, we find that we have the highest support for a model wherein hybridization has been happening for a very, very long time between these species, and we don't find support for a model of only 300 years of hybridization. This would suggest that hybridization is not solely caused by anthropogenic change in eastern North America, and I think changes how we should think about the conservation of the vermivora warblers. It may be true that hybridization is more common now than it used to be, but hybridization is a part of their evolutionary history that far precedes any large-scale human changes to the habitat in eastern North America. So to summarize, we find few genomic regions that differ between golden-winged and blue-winged warblers, and some of the regions that do differ are directly upstream of genes with an intuitive connection to plumage. And this similarity between the species is likely a result of historical and ongoing admixture, which we think has significant conservation implications. Okay, so part 2 is a look at asymmetric introgression of color in white wagtails. And this work is led by Georgi Semenov, who's been working on the system for many years. Ethan Link, Eric Enbody, Per Ahlstrom, and Leif Anderson have also helped. So the white wagtail system is quite different than the warblers here. This is a system of subspecies, widespread across Eurasia, that have multiple contact zones. The contact zone that I'll be talking about is between the alba subspecies, the distribution of which is indicated in red here, and the personata subspecies, the distribution of which is indicated in green. And during his PhD, Georgi sampled a 3,000 kilometer transect across this contact zone to try and understand what was going on and characterize the hybrid zone. One thing that he noticed while he was doing this work was that there's really simple segregation of head plumage in alba and personata in the alba and personata hybrid zone, which suggests that there's very simple genetic architecture to that trait. Most individuals either look like alba, pictured on the left, or personata in the middle, and the only hybrid phenotypes you find for head and neck coloration are depicted here. Either an alba-like individual on the left of the hybrid panel, a personata-like individual on the right of the hybrid panel, or a fully intermediate individual. Importantly, Georgi's work has shown that face and neck coloration are very important sexual signals, and that wagtails have strong assortative mating based on plumage of these regions alone. And it gets even more interesting. So when Georgi examined this hybrid zone, he found that the plumage cline for those important plumage patches, the head and neck, is very far displaced from the genetic ancestry cline. So if we first look at the top panel, this is showing how head plumage changes across that 3,000 kilometer transect. So on the left side of the panel, you have individuals with alba head plumage. Within the hybrid zone, you have individuals with alba plumage that transition to personata plumage. But you'll notice that if you look at the bottom panel, this is the genome-wide PC1 score. It shows that the genetic ancestry transitions 300 kilometers away from where the plumage trait changes. And this is one of the most curious patterns we can find in a hybrid zone, where a trait that we know is involved in mate choice and reproductive isolation has actually moved away from the genetic background. And so the asymmetric introgression of this trait sets the stage for us to dissect the genetic architecture of head plumage and get a better idea of why it's moving the way it is. In order to address this, we used whole genome sequencing of alba and personata males, and it revealed the genetic architecture of these subspecific differences. On the top panel, you're seeing another Manhattan plot, but here with just two tall buildings, one on chromosome 1A and one on chromosome 20. You can see that the average FST between these two species is very low. And then if we just look at the whole genome, we can't differentiate them. But if we only look at these divergent regions on chromosomes 1A and 20, we can differentiate alba from personata. When we zoom in on these regions, we find that they contain only a few genes, and they do contain genes with intuitive connections to phenotype. Interestingly, on the right panel for chromosome 20, a Goody signaling protein is directly upstream of this divergent region. What you're seeing here in black is FST and red is DXY, so measures of relative and absolute divergence, both suggesting that these regions are resistant to gene flow, and given the gene associations with them, we think connected to phenotype. When you look at the combinations of phenotypes that individuals have, it seems like the region on chromosome 20 plays a stronger role in dictating whether an individual has a white or black face and neck. And really interestingly, if we look at simulations of a reproductive trait like plumage, a two-locus trait with strong assortative mating and partially dominant inheritance, we actually can reconstruct the movement of this plumage cline away from the genetic ancestry cline. So we think because of this simple genetic architecture, that's why we can see the cline moving the way that it does. And finally, when we use similar demographic modeling as was used for the warblers, we find that it seems like hybridization between alba and personata wagtails probably only started about 500 years ago. And interestingly, in the region that they hybridize, development really started about 500 years ago, and these birds are highly, highly associated with human-modified habitats. So it could be that because of human actions, we've created a contact zone that then allows us to understand the genetic basis of an important reproductive isolation trait. So to summarize this part of the talk, head plumage does not match wagtail genetic background. Head plumage does not match genetic background in this wagtail hybrid zone. We found two regions that differ between the subspecies, and we think that this strong positive assortative mating, combined with dominant inheritance of head plumage, results in a flux of alleles from personata to alba, which allows that pattern that we showed to exist. So the final system I want to talk about is about color and reproductive isolation. This work has been led by Sheila Turbeck, who's a grad student in my lab, and Leonardo Campagna, who is a research associate at the Cornell Lab of Ornithology, as well as Irby Lovett, who's at the Cornell Lab of Ornithology too. So Sheila was specifically interested in trying to understand whether these plumage differences matter for reproductive isolation. To do this, she worked on a widespread species, Spirophila hypoxantha, and a very uncommon species, Spirophila iberensis. Spirophila iberensis actually was only first recorded in 2001. Since it was recorded, it has increased in population size, but prior to 2001, no one had ever seen one. Pictured here is also a Spirophila female. Females are indistinguishable both in our visual spectrum as well as the avian visual spectrum, which includes UV, which is important. So Sheila worked in Ibarra National Park to collect individuals, sequence whole genomes, and try and characterize differences, keeping in mind that Leo had never characterized differences between Spirophila iberensis and any other species. But like what Leo had found, Sheila found that Spirophila hypoxantha and iberensis differ in only 3 places in the genome. Very low mean FST, 0.0006. None of the differences between them are fixed, and only 42 regions of the genome have FST higher than 0.85. Interestingly, two of these regions are also near genes involved in the melanogenesis pathway, which is consistent with results that Leo found in his previous work. But what Sheila was really interested in was understanding whether these genomic regions are relevant for reproductive isolation. Keep in mind the females do not look different, and no one had ever looked at reproductive isolation in the field in this system, and actually in many systems it's really hard because you've got to get out there, sample nests, and so on. So Sheila conducted some really incredibly difficult field work in Ibarra National Park between 2016 and 2018, wherein she sampled 127 nests, 126 adults, and 80 nestlings. It's hard to overstate how hard it is to find the nests There's a lot of nest predation, so even if you find the nests, they might disappear before you can sample the chicks. And so this really was an amazing amount of work, and it really paid off. Sheila found by sampling pairs that there's complete assortative mating across seasons, despite the fact that there's high extra pair mating. So within a species, there can be extra pair mating as high as 37%, and an absence of temporal or spatial barriers to reproduction. These birds initiate nesting at the same time of year, and they nest within the same habitat. And I'll show you a picture of how close their territories are in a minute. So this complete assortative mating was consistent in terms of the social mates as well as the extra pair mates, and all of the offspring in a given nest clustered with their own species. So to find complete assortative mating in this, where there's a potential for hybridization, and we know these birds are really, really closely related, was pretty incredible. If you look at the habitat again, you can see it's relatively featureless, and the map on the right depicts range centers for different breeding males. And you can see that they really do overlap and abut one another, which would allow females potentially to mate with males of other species. Sheila wanted to further try and understand what signals were really important for reproductive isolation in the system, since it seems like there's really strong pre-zygotic reproductive isolation. To do that, she used mounts and playback experiment. And you can see in this image that Hypoxantha males respond aggressively and strongly to models that are the right color and singing the right song. The same is true for Iberensis males. They have the strongest responses when plumage and song are conspecific, and their aggressive responsive decline as you present them with heterospecific individuals. So it's clear that plumage and song matter for interspecific aggression and likely mate choice. Importantly, in captivity, there's no evidence of F1 inviability in crosses, so there's the potential for hybridization to occur in nature, but it seems like these prezygotic reproductive isolation mechanisms are strong enough to prevent it from happening. And so this is an interesting system because there's very little genetic variation. There's a lot of opportunity potentially for hybridization, but it doesn't seem to be happening. And importantly, it could be that Iberensis is a newly generated morph, maybe through rare hybridization of other species, that has quickly attained reproductive isolation and has an expanding population. So to summarize this part of the talk, divergence peaks underlie differences in male plumage patterning for Spirophila. Complete assortative mating by divergent genomic regions was documented by Sheila's sampling of many nests and breeding pairs. And it seems like male song and plumage pattern mediate assortative mating and appear sufficient to maintain species boundaries early in divergence, which is really exciting. So we are studying other plumage of other birds in our lab. Eric Funk is a PhD student in my lab studying the genetic basis of plumage variation in rosy finches, which are high elevation specialists in Western North America. And he's finding regions of the genome that might relate to cheek coloration, and he's continuing this work in collaboration with Kristen Ruig at CSU, Erica Zavalita, Kevin Winker, and Garth Spellman. In collaboration with Nicholas Mason, Eric is also working on the genetic basis of plumage variation in redpoll finches, which seems like it might be linked to a supergene, an inverted region of one of the chromosomes. More on that in a few months, hopefully. So by studying populations with weak or moderate divergence, we've been able to isolate regions of the genome that might underlie plumage variation, which is subsequently important for the divergence process. But like I said at the beginning of the talk, a lot of you know about my work, potentially because of our work on chickadees and reproductive isolation, where we're more interested in understanding why hybrids don't do well. And so in order to explore this, we recently were funded by NSF to study metabolic breakdown and performance in hybrid black-capped and Carolina chickadees. This is work in collaboration with Zach Chevron, Amber Rice, and Matt Carling. Amber and I, as well as Tim Roth, were also awarded another NSF grant to investigate cognitive breakdown in black-capped and Carolina chickadee hybrids, where the hybrids appear to be cognitively impaired. And as cache hoarding birds, if you're cognitively impaired, that may result in strong selection pressures against you during the winter. We're also exploring hybridization between black-capped and mountain chickadees, which we think is disturbance mediated. It tends to occur in regions humans have modified. This work is led by my student Catherine Grabenstein, who's been integral in establishing our local field site, the Boulder Chickadee Study, which now consists of 360 some nest boxes across a 6,000-foot elevation gradient, which allows us to monitor interactions between black-capped and mountain chickadees to better understand what causes hybridization to happen between them. And it's been really fun to set up a long-term study. So I hope this talk has convinced you that hybridization and hybrid zones are natural experiments that can help us understand both the genetic basis of phenotype, which was the focus of this talk in the case of the wagtails and the warblers, but also species barriers in the case of Spirophila and then the chickadees, which I'm not talking about in depth. And so I think my lab's work is kind of getting at this question, what generates and maintains biological diversity. Certainly we won't ever have one answer, but we're starting to understand from various avian systems, either weakly divergent systems or strongly divergent systems, the genetic basis of traits we know are involved in reproductive isolation, or the genetic basis of things that we know lead to low hybrid fitness. And we're able to accomplish this because we are working on multiple systems across this speciation continuum. So with that, I'd like to thank my lab, my postdocs, grad students, and undergrads, my PhD advisor, Vicky Friesen and Tim Burt, my postdoctoral advisor, Irby Lovett, and his group, the Fuller Evolutionary Biology program, as well as mentors and friends and colleagues that have helped me along the way. I'd like to acknowledge my funding from NSF, both from the iOS and DEB groups, as well as the Cornell Lab of Ornithology, National Geographic, and the University of Colorado, Boulder. So I'll leave a summary slide up here at the end of the talk, and unfortunately I'm not there to answer questions, but if you have questions, feel free to email me. My email is here at the bottom of the screen, or send me a tweet at Dr. Scott Taylor. Thanks again so much for inviting me. It really is a pleasure to be involved in the Ontario Ecology, Ethology, and Evolution Colloquium, and I hope I get to see some of you in person once things are back to normal.